Mark Rogers, you're next for the status of invasive carps in the Tennessee and Cumberland River basins.
All right, I think I'm going to go with you. Can you hear me clearly?
Yes, for now.
Okay. For now, I'm going to go with the next slide approach, and try to get done so you guys can get the last talk in and get lunch. So I'm going to take a little bit higher level approach to this, but then I'm going to highlight some other intense research projects. So go ahead and go to the next slide. Just to back up a little bit, when we talk about invasive carps, we're talking about 4 species: black carp, grass carp, silver carp, and bighead carp. They were brought to the U.S. For water treatment. And
which slide are you using?
You went one ahead, so I need to be on slide 2. Yeah, so they were brought here for pond management and water quality controls intentionally, and they escaped, and then they've provided self-supporting populations. So go ahead to the next slide. Okay, for the purpose of this talk, I'll be focused on big-headed carps, silver carp and black carp. And so on the left side of your screen, you see a 30-pound silver carp. On the right side of your screen, you see a 80-pound bighead carp. And I can't even see the figure legends or the picture legends, but that's okay. But that's going to be the focus of this talk, is on silver carp and bighead carp. Next slide. I don't see anything. Oh, there we go. You know what? A lot of you might know this, but the idea is that these things are pelagic nomads. They swim around in schools, and they're highly fecund. So are you able to see the slide that I'm seeing? The big-headed carp characteristics?
Yeah, I see some, yep.
Okay, so on the left picture, that's 6 pounds of silver carp eggs that were caught in the Tennessee River. And each of those eggs, to try to paint a picture of what the size they are, imagine a bag of grits. And I can't usually make that comparison when I'm giving this talk in the Northeast or somewhere, but each of those eggs could be a baby, but they're the size of a grit. So they're super highly fecund. They're highly mobile. They're filter feeders, so they're swimming around, sucking things out of the water. They're jumpers, and that's part of the things that that create human threats and tourist threats. They're competitors. We've seen in other places in the Midwest where, or the lower Mississippi, where these things get highly established and they can compete with other filter feeders such as gizzard shad or larval fish or paddlefish, and they're gear avoiders. So imagine being on the right and you're in an electrofishing boat. How are you going to net those? How are you going to get a reliable catch per unit effort and monitor abundance? Next slide. So Dr. Eggleton just talked about the lower Mississippi. This is the map that goes to Congress, the WRDA map, about population distribution in the upper Mississippi. And so if you look kind of in the middle bottom, you can see the red, and that's where the Mississippi takes a right-hand turn into the Ohio River. And then if you look in the lower right, you can see where it turns down into the Tennessee River. Unfortunately, this map does not show the Cumberland River. which is going to tear into the north end of Tennessee. But what this map is showing is where things are established and reproductive, and where things have been captured but not established, if you will. Okay, next slide, please. Okay, we're going to focus in a little bit more on what's going on in the Tennessee and Cumberland. So on your far left, those deeper, darker colors are showing reproductive established places. And if you come up to me to the top left corner, and then you see the orange, there's high abundance, undocumented reproduction. We had a lot of fish in 2015, but we have not in the Tennessee and Cumberland. So the Tennessee would be the one that's almost going at 6 o'clock, and the Tennessee would be, the Cumberland would be the one a little bit right of that. We have not documented reproduction in these systems, but as you look across this landscape, one thing I want to highlight is as you go down across the bottom, those red X's, those are individual reported or undocumented cases. So we're trying to track how these things are going, and we call that the leading edge, although some people don't like that term. But that kind of gives you the landscape of the Tennessee and Cumberland Rivers. Next slide. Okay, there are established management plans, and there's federal management plans and state management plans, and to try to put those excuse me, into a common framework, there are control goals, and one of them are to stop population expansion into places where they're not established and reproductive. And another one is to reduce the abundance in places where they are established. Next slide. So one of the things that state agencies along the Tennessee Cumberland, and that's 5 states, that's Kentucky, Tennessee, Mississippi, Alabama, and Georgia is kind of in there too. They want to prohibit movement by people. So in inhabited waters, these signs are posted where you cannot move bait fish from one place to another. And one of the things about these juvenile bait fish size invasive carps is they look a lot like a threadfin shad, a gizzard shad, not so much like a skipjack, but that's one effort that they can do is prohibit bait movement. Next slide. They are investing in strategically removing carp, and I'll talk about that. Next slide. So Tennessee and Kentucky are working on incentives to try to get people to remove, commercial fishers to remove these fish. And so you can see the number of pounds removed on the left axis by year. And one of the challenges to this is marketing and creating a value for commercial fish removals. And so both Tennessee and Kentucky are providing extra cost, or extra pay, I should say, to commercial fishers for trying to catch these things and get them out of there. And that takes a lot of work. Commercial fishers, they can go fish for catfish, they can go fish for smallmouth buffalo or other commercial fisheries. You have to make it worth their time. And you can see kind of a response there when And a lot of this is due to federal aid that increases the capacity to remove. Next slide. Another thing is part of the control strategy is to install barriers to stop or reduce upstream movements and try to be able to figure out how to, and Alan Brown's in the room, he could tell you about his experience on this. But next slide. We want to stop them moving upstream to places that they aren't established. So we went through a structured decision-making process, and this shows you the map before, but I want you to focus on the pink circles. Multiple state fisheries people weighed in on this, and this was kind of the take-home of, if we want to stop these things, where would we put barriers? And we've worked with TVA, USGS, Fish and Wildlife Service, all the states that are invested in this, and saying, if we had the money, where would we put barriers to at least impede movements from upstream and impede established populations? So just looking at this, the connection in the upper left, there already is one at Barkley, and hopefully it's still kind of in a test phase. And Alan could tell you more about the longevity of that. But because we still feel like they're coming in from the Ohio River, Kentucky Dam on the Tennessee River would be a natural place, or would be a priority place to put a barrier and try to impede that inclusion into it. If you look down at your bottom left, Pickwick, that connects to Bay Springs and then to the Mobile Basin, and that's another high priority. And then there's other places that have been thought about based on locations and just what we think will do the best effect, but it'll all have to be tested. Next slide. And then the last thing that we're doing is we're monitoring abundance of movements, and that's what I'm going to talk about. So next slide. One of the things that we're doing is we're trying to figure out how these fish move around. And this is a multi-state, multi-university, multi-agency cooperation where we're using acoustic telemetry to try to figure out where these things are going, when they're coming through the dams, because we cannot impede barge traffic because they have to go through up and down the river. But at the same time, we're trying to balance that with not letting carp through. Next slide. So right now, if you see the little black dots on this map, then that shows you where we have all the telemetry receivers that are tracking where these fish are moving. And the big kind of darker ones, we have all the dams and all the locks on the Kentucky River, which is the one north at the top of your screen, up to Old Hickory Dam. And then on the south end, we have all the locks and dams to Gunnersville. And we're trying to track every fish that can move through. And so that tells us, is there maybe a time of season, time of day? When are these fish using this to make upstream passage? Next slide. So when we talk about the importance of monitoring and research, we want to know what the status is. What is the size structure? What's the age structure? We want to know, are the incentives and all this money going towards commercial fishing? Are they removing enough fish to have an impact to where it's not going to affect native fish? How and when, and this is the telemetry stuff I was just talking about, how and when are they using these locks to go through? What barrier types are most effective? And that's more of an experimental framework. I bolded here, how are other species affected? And so let's go to the next slide. I'm already at 15 minutes, so I might have to cut this a little short, but let's go ahead to the next slide. And I'm going to tell you about a project, and this was funded through the Small Grants Program. And we wanted to know about what are silver carp going to do to our freshwater mussels. So you can go ahead. And again, this is through the Small Grants Program. This was, I don't know, Cindy might remember, maybe a $15,000 project. I want to focus on a tributary to the Tennessee River, the Duck River. And I want you to, yeah, stay there. The reason to focus on the Duck River is when the Nature Conservancy says you have the most diversity of anywhere in North America, that's a concern when there's an invasive species. It's one of the 3 most diverse mussel hotspots. It has 650 riverine fauna. You can read the rest. Critical habitat, according to U.S. Fish and Wildlife Service, for 2 endangered mussels. So why would we care about silver carp? And that's me in the lower right picture. That's me with the silver carp in the Duck River. And behind me is the Columbia Dam. The Columbia Dam is about 10 foot high. And so it could be a movement barrier, but let's carry on to the next slide. If you remember about the life history of native mussels, and I just told you that USGS said this is one of the 3 hotspots for mussels, they have to have a host fish to carry their young Glochidia, and then they settle from there. But at the same time, these mussels have to filter feed. That's where they get their food. And if you remember from one of my introductory slides, silver carp are filter feeders. So the concern is if silver carp are sucking things out of the water on their gills that mussels need, then that could be a threat. Next slide. So the purpose of this project was to look at overlap between silver carp and native mussels. And on the bottom right, that's a gill raker out of a bighead carp. But you can see that you have a lot of filaments that can suck things out of the water, whereas a mussel just has a straw, basically, and it proposes a threat. The other thing we wanted to do is use eDNA techniques, and that was talked about before, of trying to confirm presence of silver carp in this watershed, or how far upriver. Next slide. Okay, all right. Yeah, I want to keep you on track to get to lunch. So what we did, because the concern was, what are mussels feeding on? What are silver carp feeding on? And this is a little bit technical, but we basically took 3 mussel species that have different host fish, and we wanted to know, what are the carp feeding on? What are the mussels feeding on? So next slide. And so this was a stable isotope analysis. In this slide, you can see that we collected everything we could. This is number of samples, and we just wanted to know how much overlap could there be in energy going to mussels versus carp. Next slide. Well, I mentioned the Columbia Dam, and in terms of what the mussels were using, there was no difference. At this time, there was no silver carp above the dam. So above the dam, we got a baseline. Below the dam, we had overlap. We didn't see anything about seasonality, but we had to pull the data across the seasons because the carp were still at such low abundance that I think the previous slide said that we got 23, and we couldn't separate it by season. Next slide. This one is, so it's a stable isotope analysis, and I want to start you on the right side, and what you should see there is on the X, we ended up combining groups and trying to see what that D15 nitrogen level is telling us is what trophic level they're feeding at. So you see high overlap between the mussels and the carp. Everything else is separated. So they are feeding at the same trophic level, which makes sense because they're both filter feeders. But if you look on the left side, what that carbon is telling us is where they're getting their nutrients from and what's providing their growth. And that one, we see that the carp and mussels are separated. So when you, the take-home message is, and this was kind of a pilot study, and but I think has strong implications for the broader needs of food web studies on carp and native species. Where they get their resources from are different, but they're feeding at the same trophic level. Next slide, please. All right, you can speed through these. Okay, so they're feeding at the same trophic level. They're both sucking out plankton from the water, but they're getting their plankton from different places. All three of the mussels, and we used common mussel species because we had to sacrifice them for a stable isotope, so we weren't using threatened, endangered, or anything. But there was no difference between the three that we used of where they're getting their nutrients from. We were doing eDNA, and there was not any silver carp over the Columbia Dam yet. Next slide. Okay, so here's the heartbreaker. In 2018, we got positive eDNA signals. We had high flows, record floods, and the dam was flooded, and we started getting positive eDNA signals, and then TWRA went out and caught this fish above the dam. So yeah, that kind of stunk. I still have, I think, I'll refer to or defer to the moderator. Do I still have 5 minutes?
Yeah, maybe not for questions, but yeah, we have about 7 minutes for till 11:30.
Okay, so we were able to pick up DNA from eDNA from the water above the dam, and then TWRA was able to confirm by putting a fish in hand. And that's one of the things is with eDNA, you can get a signal, but when you see the fish in hand, that's a pretty confident conferral. Next slide. So I'm gonna go through this quickly. We still have questions about what these carp could do in these large reservoirs that are different than the Midwestern rivers that have been studied or the lower Mississippi. Next slide. There you go. So we wanted to look at the availability of planktonic particles in the water column. We wanted to look at what big-headed carps were selecting for, or what they, I can't say selecting for, but what was coming into their gill rakers versus what was going into gizzard shad gill rakers, and then assess the overlap. Next. So we worked in Barkley and Kentucky reservoirs, which are the most downstream reservoirs in the Kentucky and Cumberland Rivers. We would catch a fish, we'd remove gill rakers, we'd rinse everything that was in the gill rakers, because remember they're planktonic, planktivores, and then we would take a tube sampler and see what was in the water. Next slide. We used a flow cam which can sort particles by size and type. So it can tell you the dimensions of things. It can also tell you, it can assign what the what type of plankton it was. Next. This is the big tank hole. What you have here is a graph of area-based diameter. So that's kind of the roundness of the particle for big-headed carp versus gizzard shad. And what you see here is a difference. This is classic Ancova, where we see different uptake of particles by season. Next. So kind of the results here, gizzard shad always selected for larger particles in every season. And that was really surprising, because if you know what a gizzard shad is, it's kind of a small fish, less than 8 inches, whereas big-headed carps can be, 20, 30 inches and have bigger gill raker size. One thing about that is that big-headed carp could filter larger sizes, but they tended not to. Next. One of the things is this big difference in the spring when all the other sport fish and other things are hatching, and every fish eats plankton. Unless I'm wrong, Dr. Eggleton, every fish eats plankton at some time, and most of them are born in the spring. The fact that that spring was our biggest effect might suggest that these other fish, crappie and things, could have successful spawning. And then it helps us understand what these things are doing in the Tennessee and Cumberland Rivers and these large reservoirs where they've never been evaluated. Next slide. So this picture was taken below Barkley Dam, I believe, and that's the invasion front that we're confronting. We are trying to figure out how these things might affect large reservoirs that have never really been evaluated on the scale of a Kentucky or Barkley Lake or a Pickwick or something like that. These things are knocking on the door. There are control methods in place. There's discussion and already been structured decision making about barriers, and then there's ecological research going on. So with that, I'm done. I thank you.
All right, thank you, Mark. We have a good time for a question, if anybody has any. I thought it was really interesting. Yeah, I don't want to go into too many questions, but appreciate your time, and sorry for the technical difficulties again.
No worries. And I think I probably need to do an acknowledgement. Working with the state agencies, the federal agency, MICRA, the MICRA has been a huge impact on this, but also the small grants initiative. We're just trying to do everything we can to figure it out.
All right, thanks.